Welcome to Tiny Expeditions, Season 6, Episode 4. I'm Dr. Sarah Sharman, here to help you understand the science.
And I'm your co-host, Chris Powell, and we are talking about brain health. We're getting our brains all good and healthy this season. And that's kind of a heavy topic if you really sit and think about not just what makes our brains healthy, but what happens when things go wrong in our brains. And that's kind of where we've been living the past few episodes.
Yeah, for the past few episodes, we've been talking about some of the more rare brain diseases, but today we're going to hit one that affects a whole lot more people, and that is neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS.
Okay, so I have a little bit of insight here on this. I know what's going to happen because I'm from the future, but where we're going with this episode is we are going to talk about those diseases, but we're going to take a little bit of a different angle at getting to those diseases, correct? We're not just going to talk about Alzheimer's. We're going to talk about ways to possibly treat and some headlines that you may have seen that have popped up that give us a bit of hope in the battle against Alzheimer's.
That's right. So you've probably been seeing Alzheimer's and other dementias and diseases like Parkinson's in the news a lot lately, partially because they've gotten kind of caught up in the political stuff we're not going to talk about on this episode, but also because there have been a lot of really exciting discoveries. So some drugs that might slow down these diseases, and even some blood-based tests that can detect these diseases maybe before you have the worst of the symptoms. So that's really exciting stuff.
To think about the idea that you can walk inside of a doctor's office or a clinic, have a blood draw, and be able to determine your risk for some of these neurodegenerative diseases. I mean, that's a game changer for us.
And we're lucky here at HudsonAlpha in North Alabama to have some scientists who are working on some of that baseline knowledge that is feeding forward into these diagnostics and these treatments.
So let's meet them now. Here are two researchers that you will hear from throughout the episode, but we're going to go ahead and introduce you to them now.
So my name's Ben Henderson. I'm a senior scientist in Rick Meyer's lab here at HudsonAlpha, and my area of expertise is I look for genomic biomarkers of neurodegenerative diseases such as ALS and Parkinson's disease.
Bri Rogers. I'm a postdoc in Nick Cochran's lab here, and I study neurogenomics. So basically, we look at understanding how genes that are associated with dementias are controlled.
We're excited for you to get to hear from these scientists today. They've got a lot of great stuff to say, but before we get to what they have to say, let's kind of reset and define some terms. Now, we're talking neurodegenerative today. We've kind of gone back and forth between that, neurodevelopmental. So help us locate ourselves. What are we talking about with neurodegenerative disease, Sarah?
Sure. So neurodegenerative diseases generally occur later in life, and they happen because you get damage to cells in your brain, your neurons. So this happens generally because you have proteins that aggregate and cause clumps in your brain cells, and that causes irreversible damage that kind of builds up over time. So they're very progressive diseases.
And just once again, the examples for these would be Alzheimer's and
Parkinson's disease, ALS, dementias.
Okay, and as we shift to Dr. Ben Henderson, he's going to talk to us about biomarkers. So give us a brief overview of that. What's a biomarker?
Sure. So a biomarker includes the blood-based detection I talked about earlier in the introduction, but these are just basic measurements that you take of the human body that can tell you things. It tells you if you're at baseline or if you've deviated from baseline. So an easy one that a lot of people don't realize is a biomarker is your blood pressure. So your doctor can measure blood pressure and tell if you've deviated from the baseline of where it could be. And usually biomarkers are used to tell like health between health and disease.
We specifically, when we think about this, I guess, the ones that are very popular right now are these protein biomarkers, such as like p-tau217, amyloid beta, a lot of the different tests that are coming out right now for those that are really great. So these are allowing us to identify disease earlier. They're allowing us to subtype disease a little bit better, which is really big for enrolling people in clinical trials. And it's also allowing us to classify between different diseases really well. So it's really important for neurodegeneration because it's a pretty long diagnostic odyssey for a lot of these diseases. There's a lot of work that the neurologists have to put in because, I mean, some of it's still a little bit of, art, diagnostic art when it comes to trying to figure out the right disease that someone has. And so if we have these biomarkers, we can actually diagnose people better now.
Thanks for helping us set the baseline of why these biomarkers are important in neurodegenerative diseases. Now, if you could take us a little bit more specific and maybe explain to us what we're doing here at HudsonAlpha in the biomarker space.
There's a little bit of, I guess, history to go back to begin the answer here. So in 2018, Rick's lab, so Rick Meyer's lab, had a great study that they published about looking at small RNAs as biomarkers of colorectal adenoma, which they found like certain species were very predictive of whether or not someone could have a cancerous polyp, something like that. And so when I started here, my main question, so I started as a postdoc before becoming a senior scientist in Rick's lab. I was really interested, could we take that technology that was used there and could we move that into neurodegenerative diseases? So that's what we've been focused on doing for like the last 6 years. Specifically right now, we are looking at ALS and Parkinson's disease. So the ALS project is a little bit further along just because the nature of when we started collections and when that project started. And we just started the Parkinson's project. I guess a year ago is about when we started enrolling patients, a little bit over a year ago, year and 2 or 3 months, started collecting samples from patients. But what we're focused on is looking at small RNAs. So we think about DNA, RNA, protein as the central dogma of molecular biology. Well, there's another type of RNA called a small RNA, and that is just because they are physically small. They're 22 nucleotides in length, and they help regulate a bunch of different biological processes. So they're really important in things that happen in disease or just in the cell in general. So we measure those agnostically from patient samples. So we worked with the Crestwood Medical Center, their ALS care clinic, which is a center of excellence designated by the ALS Association. We worked with them, and Sherry Kolajajak is the nurse there who helped run this, to enroll patients there to be part of this study. So we took a blood draw from those patients, brought it back to the clinic. I spun it down, got the plasma out, and then we had this long process to do the sequencing and then the analysis from the sequencing. What we saw is looking agnostically at all of these small RNAs, and then not just taking our data, but another group's data that had previously published a similar study. We found that we could settle on one species in particular to distinguish ALS from controls.
So let's put this into the real world. Let's say I am going to the clinic to get a test. What does that look like from just walking into the clinic to walking out? What can I expect?
So what it would look like is it'd be a simple blood draw, right? They go in, they give blood, blood is spun down, plasma is taken away, and then it's like a regular kind of qPCR clinical test where it's sent off to a lab and it's the analysis is performed, and they get back basically like a score of where they are on the ranking of how much they have of this biomarker, and that can really kind of tell do you really have ALS, or hopefully we can subtype disease a little more based on that in the future. But I think what it could also look like too, and where it actually could have really good utility, is in combination with potential therapeutics. And not to say that like, oh, measuring this is going to make you better. I don't mean it like that. I mean that because the biomarker we saw is really tied to biological processes that are happening in ALS, that as people get treatments and then have this biomarker measured along the way, you might actually be able to see a decrease in that biomarker expression as they get treatment. So I think there could be a lot of power there as well.
HudsonAlpha is obviously a nonprofit, but I know also a lot of academic institutes work on this. Are large companies doing a lot of the baseline research, or is that kind of fall to the academic researchers to provide with them with those biomarkers that would end up in a test?
No, I mean, a lot of the basic research is done by academia, right? I mean, when we think of let's look at the Alzheimer's biomarkers, for instance, amyloid beta and tau. That is decades upon decades of academic research that established a relationship with amyloid beta and tau for Alzheimer's disease as these pathological hallmarks of disease. And then academic labs then saying, hey, we can measure it in the blood, and then developing an assay and then passing it on. So yeah, industry really relies on academia to do a lot of the base work, which is important and why we need the funding for that. Yeah.
Let's shift gears here for a second. We talked to Dr. Ben Henderson about the possibilities and what a diagnostic test would look like. Let's shift now to treatments. What would that look like if we were able to find treatments and the research that is getting us to the point of finding those treatments? And we're going to talk with Dr. Bri Rogers.
So over the past couple of years, in the news, there have been a lot of announcements of new treatments for some of these dementias. What role does genetics play in getting to a treatment like this?
Yeah, so a lot of the drugs that you hear about today are anti-amyloid antibody therapies, right? And so much of the research that led to that was trying to understand what's called the amyloid cascade hypothesis. And this is basically just saying that the accumulation of amyloid beta into those plaques that are pathological in Alzheimer's is early, and it's like central to Alzheimer's disease development and then progression. So really some of the first genetic links to Alzheimer's disease have come from studying that pathway and trying to figure out why that happens. So there's families that have early onset Alzheimer's disease, like throughout their family. And so studying those, we found these they're very rare, but they have a big impact on basically causing early onset Alzheimer's disease. And these are in genes called the amyloid precursor protein, or APP, and presenilin 1 and 2. So these basically affect how amyloid is processed. And it so they affect how that is turned into the longer form of amyloid beta. So that longer form is more likely to aggregate, and then that leads to those plaques. You may have also heard about APOE, E4, and understanding your risk for late onset Alzheimer's disease. And that's also involved in amyloid. So APOE is actually involved in clearance. And so normally it's going to clear out those plaques that would build up. But if you have the E4 allele, it's not as effective. So then you get much more accumulation.
So you talked about some early discoveries in folks who have early onset disease and folks who have late onset disease. What are you doing here at HudsonAlpha to kind of understand the genetics even before that to all the way to the disease?
Yeah, so those variants that I talked about are really rare in the population. So what I spend a lot of my time looking at is more common variants. So these are more prevalent in just the general population. And these have smaller contributions to disease risk. So like I said, if you have those rare mutations of those other genes, it's almost near certain that you would get early onset. But these is so much, just because you have that variant doesn't mean you're going to get it. So it's more likely that these variants are kind of working in combination. And so it could be several variants in several different genes that's basically pushing your disease burden over a threshold. So what I try to understand is identifying those variants and which ones are important.
So when you're talking about identifying these variants and you're looking at the common, it sounds like when you're dealing with multifactorial data like that, it gets very complicated very quickly, right? Mm-hmm. So what are the challenges that you face in trying to link these variants over and say, what's actually happening? How does this increase risk? What are some of the primary challenges in looking at the common variants?
Yeah, so a lot of times you'll hear us say disease-associated variants. And that's because these variants are they're just correlated with disease. We've done these large sequencing studies, and we're basically saying in people that have the disease, this variant occurs more often, it's associated. So actually finding what the causative variants are is really hard. And that can be because the majority of the variants that we're seeing that are disease associated are in non-coding regions of the genome. So those are regions of the genome where there's not a gene there. Genes are what give the instructions to make proteins that actually do the stuff. So if there's not a gene there, we don't really know what the target gene is of that variant. So most of these are affecting regulatory elements, and those control how much of a gene is made. So genes can be expressed at different times at different amounts, and these regions are what's controlling that. So basically, that also makes it hard, because when you're trying to model that in an experiment and say, no, like we took this variant and now we're looking at it in a cell culture experiment, well, if you don't do that at the right time or in the right cells, you're not going to see the effect. And so that's been something that's been more difficult in the past, as we've done a lot of experiments with like cancer cell lines. That's been the easiest to work with. But now that we have these models where we can make human brain cells in a dish, it's a lot easier, but those are a lot harder to grow too.
Right.
So there's a lot that goes into it, trying to figure out what an actual causative variant is.
So when it comes to HudsonAlpha, is there anything that you can highlight for us? Anything that's specifically genetics focused, that's a project related to Alzheimer's or neurodegeneration that's happening right now that you're working with?
Yeah, so a lot of what we're talking about, a lot about what you see in the news is all about amyloid beta. But in Alzheimer's disease, there's also tau tangles. So that's the other big pathological hallmark that you see. And there are clinical trials going on aimed at reducing tau burden in the brain, and those show pretty promising results. We're not to like FDA approval or anything yet, but it lets us know that like that may be an important step, that maybe we even have like combination therapies. So one project that I've led has been understanding how the gene that makes tau, called MAPT, is regulated. So we wanted to understand what are the regulators of MAPT, identify those, and look at them in the right cell context. So we found that there were like a handful of what we call enhancers. And so they're just controlling how much tau is made. You can think of that like a light switch dimmer instead of a switch. So it's not on off. It's just controlling the levels of it that are made. And so basically we found these and saw that some were really only working in neurons. And those are the cells that are most affected by tau tangles in Alzheimer's. So that was really interesting. It's pretty cool. We published that about a year ago, but it's actually laid the groundwork for how we're kind of going after other genes that are really important for different neurodegenerative diseases like Parkinson's.
I don't know about you, Chris, but I'm just so excited and feeling just really hopeful after hearing all of the really cool discoveries happening here at HudsonAlpha. They're closely related to things that are happening in the news.
That is true, and there's so much good stuff. What I think we should do is pause for a moment and go back, do a TLDR. Let's go back and look at what we've learned today.
Sure, let's do it. I'm going to sound like a broken record because I think I've said this on several episodes now, but for me, what stuck out is just how crucial basic science is for uncovering the knowledge base that eventually leads to these breakthrough treatments and diagnosis. And I mean, it's especially important right now while we're sitting here in 2025 that people really understand that, because as, funding shifts, we still need people like giving through philanthropy for things like basic science research.
Yeah, and one of the things that stuck out to me is to go back to Dr. Ben Henderson when he was talking about biomarkers and the possibility of early testing. That, to me, is very, very hopeful, because as of right now, many of these neurodegenerative diseases, you don't know until you are full on onset, right? Mm-hmm. And so being able to have early detection and non-invasive early detection is a game changer to me. You know me. You know that one of my goals in life is to have as much information about myself as possible, almost like a gamification, right? The more data you have, that changes how you live your life. And I think that's really important. And the fact that this could be really accessible, non-invasive, and the fact that it's a reality, right? This isn't science fiction. It's here and it's available. And I do want to throw out a word of caution here, and you can go back and listen to, I believe Dr. Rick Myers talking about this, but with that knowledge comes great power, right? And so we have to be careful how we use it, and we have to think through whether or not we want that info. But if it is something that we want, having it available is a game changer.
And on that note, that's a wrap on episode 4. Thank you for joining us for this episode about neurodegenerative diseases in the news. We hope you learned a lot about your brain and the research being done in brain health.
We'll be back again for the season's grand finale, and we'll introduce you to the emerging field of phenomics and discuss how it is helping us build a 360-degree view of brain health.
Tiny Expeditions is a podcast about genetics, DNA, and inheritance from the HudsonAlpha Institute for Biotechnology. We're a nonprofit research institution in Huntsville, Alabama, with a unique mission.
We bring together scientists and companies to develop and apply genomic advances to make a better world. That includes everything from brain research to agriculture for a changing climate.
If you enjoyed this episode, swing by your favorite podcast app and hit that subscribe button. And don't forget, we're also on YouTube. While you're there, consider leaving us a review. It really helps us spread the knowledge.
Thanks for joining us.